DNA molecules are subject to several topological constraints. In this video, we will define the parameters that describe the topological constraints. These parameters are the linking number, also called LK, the twist number, also called TW, and the writhing number, also called WR. We will also define the relationships between these three parameters. Genomic DNA exists either as a single circular molecule, as found in the genome of most bacteria, or as a series of linear molecules, like in human chromosomes. The relative length of a DNA molecule implies that any changes in the structure of the DNA double helix result in topological constraints. This is partly because the DNA double helix is packed into higher orders of structure. Circular molecules can adapt to conformational changes. Here we see a fully relaxed DNA molecule and a supercoiled DNA that is characterized by a compact twisted formation. Supercoiling is the consequence of topological constraints that are applied to a molecule with no free ends. Different levels of supercoiling exist, depending on the nature and extent of these constraints. But what about linear DNA molecules found in chromosomes? They are also subjected to the same topological constraints as circular DNA molecules. While linear DNA molecules are free, they are simply too long to ease constraints by free rotation of one strand around the other. These molecules are considered long double helices with tethered ends, and linear DNA molecules also form supercoiled structures. Topological constraints arise from the fact that the two DNA strands repeatedly intertwine. Untangling the two strands of the DNA double helix occurs during major biological processes. These include the opening of the double helix at the site of DNA replication, the separation of two copies of the double-stranded DNA molecule at the end of replication, and the localized opening of the double helix during transcription. Unwinding a DNA double helix requires the free rotation of the ends of the DNA monomer, which can be easily achieved for very short molecules in solution, but is challenging for long linear DNA molecules found in eukaryotic chromosomes. In eukaryotic chromosomes, free rotations are restricted due to bound proteins. And in circular DNA molecules, free rotation is impossible due to the absence of free ends. A topological domain is a fragment or stretch of DNA double helix for which free rotation of its ends is impossible. For example, the circular genome of a bacterium is considered a topological domain. DNA loops that are attached to the protein backbone of chromosomes are also topological domains, as well as linear molecules with both ends attached to a membrane, as is the case with some viral genomes. A segment of DNA that is limited by two large protein complexes also constitutes a topological domain. The consequences of a partial strand separation of a topological domain can be illustrated with a simple example, such as a tangled phone cord. The phone cord is like a twisted strand between two fixed points, the receiver and the phone. The twist of the cord tightens or loosens between these fixed points as a consequence of moving the phone or receiver randomly. The result of this constrained movement is annoying supercoiling of the phone cord. Similarly, in the circular molecule of DNA, topological constraints are created by either the opening or the closing of a portion of the DNA double helix. This constraint can be relieved by DNA supercoiling when the DNA strands are not broken, or by breaking one or both DNA strands. The topological state of DNA is defined by three parameters: the linking number, the twist number, and the writhing number. The linking number is a topological property that describes the total number of times one strand of the DNA double helix wraps around the other. It is an integer that remains constant as long as the DNA molecule is not broken. This can be visualized as the red strand crossing the gray surface, which is encircled by the green strand in the diagram. To change the linking number, a three-step process that is catalyzed by specific enzymes is required. One strand has to be broken, the two strands rotate relative to each other, and then the break has to be repaired. The linking number is always an integer. The twist number represents the total number of times the two strands twist around each other along the helical axis. For example, if a circular DNA molecule has 360 base pairs and, assuming a pattern of double helix with 10 base pairs per turn, the twisting number would be 360 divided by 10, which equals 36. The twists can change without breaking any strand. In DNA, which is a right-handed helix by convention, the twist is considered positive. The writhing number is the number of times the long helix of a double helix DNA is twisting around itself, which represents the overall shape of a twisted DNA molecule. The writhing number can also change without breaking any strands and can be either positive or negative. It is always much smaller than the twist number. In the absence of strand breakage, the linking number remains constant, while the twist and writhing numbers can change. The twist and writhing numbers are interconvertible, and the sum of these numbers remains equal to the linking number. In other words, the linking number is equal to the twist number plus the writhing number. For example, in a circular molecule with 5,000 base pairs, if the twist number is 500, which is calculated with 10 base pairs per turn, there are 4 negative supercoils. Consequently, the linking number for this supercoiled circular DNA molecule will be 496. Equilibrium between supercoiled and relaxed DNA molecules can be achieved by opening the double helix in a region of the DNA. When the double helix is opened, the twist number decreases. In terms of representation, if the first base pair is positioned at the top, negative supercoils are depicted by the front double strand crossing the back double strand from right to left. In this case, there are 4 negative supercoils, and the writhing number will be -4. If the twist number changes from 500 to 496 while the linking number remains unchanged, the DNA is uncleaved, the writhing number should increase from -4 to 0. This eliminates all supercoiling and leaves the DNA in a relaxed state. Conversely, if the region of the double helix that was separated is rewound, the twist number switches back from 496 to 500. Let's delve into a few other definitions. We've discussed what the LK number represents. Now let's introduce L0, the linking number of a fully relaxed DNA molecule under physiological conditions. So, once again, considering a simple example of a 5,000 base pair molecule, the linking number is L0 equals 500 because it's fully relaxed with no supercoiling. When DNA is supercoiled, the linking number differs from L0, and the degree of supercoiling is evaluated by the difference between LK and L0, which is denoted as delta LK. By convention, if delta LK is negative, indicating that LK is lower than L0, then the DNA is said to be negatively supercoiled. Conversely, if delta LK is positive, meaning LK is larger than L0, then the DNA is positively supercoiled. Supercoiling induces bending and torsional deformations of the DNA molecule. Supercoiling is energetically unfavorable, as shown by the free energy calculation for negative supercoiling. Delta G is greater than 10 times RT times sigma squared. Regardless of the value or sign of delta LK, the superhelical density, sigma, is always squared, resulting in a positive delta G. So, under physiological conditions, why do we find slightly negatively supercoiled DNA? The reason is that negative supercoiling facilitates local changes in a twist, which aids in the relaxation of the DNA during processes such as replication and transcription. In essence, the presence of negative supercoiled DNA at physiological states promotes the efficiency of processes like replication and transcription. The topological states of our currently closed circular DNA can be understood by starting with the equation linking number is equal to the sum of the twist and writhing numbers. Circular DNA can be interconverted into supercoiled DNA without changing the linking number. Under this condition, you have to interconvert the twist and writhe. Now, is it possible to change the topology of DNA by changing the linking number? This change of linking number without a simple interconversion of the twist and writhe requires the breakage of at least one strand of the double helix. This breakage is possible and is catalyzed by enzymes called topoisomerases. There are 2 classes of topoisomerases. Class 1 breaks only a single bond and then repairs it. Class 2 breaks 2 strands of DNA and then repairs them. In our experiments, a relaxed plasmid with an LK of 500 and the topoisomerase can move the LK number from 500 to 496 and add negative supercoils without changing the twist number. In conclusion, the topological constraints imposed upon the DNA double helix are the consequences of intertwined strands in the double helix, the length of linear chromosomes, and the lack of free ends in circular DNA. The topological state of a DNA double helix is defined by its linking number. The linking number is the sum of the twists plus the writhing number, and unless one or two strands are cleaved, the linking number remains constant. This means that twist and writhing are interconvertible to maintain this constant linking number.